Hello and thank you for listening to the 5th season of the ISTAART PIA Relay Podcast, brought to you by Dementia Researcher. ISTAART is a professional society and part of the Alzheimer's Association, representing scientists, physicians, and other dementia professionals active in researching and understanding the causes and treatments of Alzheimer's disease and other dementias. In this 6-part series, we have members of ISTAART Professional Interest Areas taking turns at interviewing their colleagues and being interviewed themselves, with the interviewee going on to be the next episode's interviewer. We're sure you've listened before, so you know what to expect. We'll be releasing one of these podcasts each day in the build-up to the Alzheimer's Association International Conference, which this year takes place in Philadelphia. Through the series, we hope you'll learn more about some of the amazing research happening in different fields of discovery and about the great work taking place in the ISTAART PIAs. Thank you for listening.
I'm Arlene Astell. I'm a professor of cyberpsychology at Northumbria University in the UK, and I'm also director of the Dementia Aging Technology Engagement Lab, or the DATE Lab, at the University of Toronto in Canada. I'm currently chair of the Non-Pharmacological Interventions PIA. And today I am delighted to be talking with Caitlyn Fastenau, and let's see what she has to tell us.
Thank you for having me. I'm really excited to have this conversation to talk about the intersection of different PIAs and the activities that we're doing. A little bit about myself. I'm Caitlyn Fastenau. I'm entering my 5th year of my PhD in Integrated Biomedical Sciences with a focus on biology of aging. And my research focus is based on neuroscience. And I kind of approach that from a translational perspective, which I really have enjoyed. I work in mouse model systems as well as cell culture, primarily looking at the innate immune cell in the brain, microglia, and that links really nicely to my work on the Immunity and Neurodegeneration PIA. And then the flip side of my research, kind of along that translational perspective, is that I work in understanding mechanisms of Alzheimer's and other neurodegenerative diseases in postmortem human disease tissue, essentially. So I work at our university's Alzheimer's Disease Research Center, or an ADRC, and we have a brain bank, and my faculty advisor is the co-director of that brain bank. So my thesis research has benefited immensely from being able to have access to cases and to probe various proteins as well as modifications that I'm particularly interested having to do with innate immune activation in Alzheimer's disease.
Great. Well, there's a lot to unpack there, which hopefully we will have time to go into. But I just wonder if we can sort of step back a bit and if you can tell us what brought you into dementia research.
That's a fantastic question. So really my journey in understanding the mechanisms of aging started when I was a child. My grandparents we lived in a multi-generational family in one home. And so I was raised very closely with my grandparents, who obviously were aging as I was aging as a young child. And I was uniquely aware of the physical as well as the cognitive kind of side effects of aging. While they didn't have dementia, thank gosh, I did see family members getting diagnosed with Alzheimer's disease and other kind of neurodegenerative disorders. And I was always a question asker. I've always been very inquisitive as a child. So I thought, okay, I'm going to kind of approach biology of disease when I went to college. And then I had an amazing opportunity to get onto a training grant called the Advancing Diversity of Aging Research. It's an NIA-funded grant, and I went to UC Davis and I did that. And honestly, opened my eyes to dementia and the immense gaps in knowledge. So then I pursued my graduate degree in it. And honestly, I love Alzheimer's disease. I'm really, sounds weird to say that. I don't love dementia because we should have a world without dementia. I agree. But I really enjoy the field of Alzheimer's researchers. It's not that I've had many fields, but I feel it's very collaborative. It's very inclusive. I think it's also very respectful of different opinions in terms of scientist perspectives and approaches to trying to answer questions, and a lot of convergence as well, which I appreciate. And as a trainee, I think I've had an opportunity to get involved in ways I wouldn't have otherwise been able to get involved and take on some, student leadership positions, which has been really unique. So I think allows students to take a little initiative in their own training journey, as well as the awesome kind of collaborative opportunities.
Oh, that's great, and it sounds as if you had to turn that childhood interest and observation of your own family into something that's become a real
Yeah.
passion, which is really important for a researcher. I think you have to be really excited about your research,
Yeah.
and then you bring other people along with you, and we can hear in your voice that you're very excited about this, which is wonderful. And I think that's also important. You mentioned that you're doing a lot of your focus on translation.
Mm-hmm.
And I wondered if you could just sort of tell us a bit more about that, because often it's seen as a separation from the sort of hard
Mm-hmm.
science, the bench science, and the science with the people
Right.
and I wonder how you approach that translation.
I think that's the age-old question. As a trained biomedical kind of basic scientist, I think that's definitely a hurdle that we have to overcome in terms of advocating that our intriguing findings in a cell culture model or in a mouse model is efficacious to investigate, non-invasively in human systems. But interestingly, I actually started in humans in preclinical testing in older adults that have MCI. So I actually worked in neuropsychological cognitive testing in my undergrad for 3 years. And I enjoyed that immensely. I'm a certified yapper. I love to talk. So I enjoyed building connections and kind of fostering community in terms of understanding what different populations in a local area were the resources to medical care and dementia care and memory centers. And then how can we screen those at a kind of, I guess, unified kind of realm in terms of we were taking into account the impact of bilingualism specifically as a cognitive scaffold to reduce your symptoms of mild cognitive impairment, essentially. So I started there and I said, wow, I love working with preclinical patients or enrolling patients in clinical trials. But I wanted to because I was often asked, okay, what's the biology behind this? And I wanted to really understand the biology, because if you're knowledgeable about what's happening, at least from what we can understand in the brain and in the body, I think you can provide that insight to patients or the community to build a better understanding of what we know and then what we need in our community participants to kind of bridge those gaps. And then in my current research as a graduate student, I haven't had the opportunity to work immensely with patients, but I work in an ADRC that obviously sees patients. So I have a lot of medical and kind of personal background in my cases. So I'm able to create correlations on cardiovascular disease and brain neuropathology, which is a really cool way. And then we take those findings and then we go to the community in public events. We say, hey, hypertension, it's a really big issue. Managing your hypertension and increasing your physical activity is good for your brain. And these are the data that support that. But I will also plug translatability and working with community participants. A huge component of that is also understanding the local population in our area and creating a diverse group of patients and population participants that represent that area. So I'm currently based in South Texas, in San Antonio, here in the United States, and where our demographics of the city are 65% Hispanic and Latino. So we really try to build that connection with our community and build trust that they will come to us and say, hey, Hispanic individuals have a 2.5 times more likelihood of developing Alzheimer's. How can we address this? How can we understand the social, the political, the demographic influences that lead to these obviously kind of differences in diagnostic rates? So I think that particularly that is my passion, is I want to understand how people from different backgrounds and different walks of life are kind of obtaining equitable access to research, to medication, to clinical trial, but also how we can provide information back to the communities, kind of like a two-way street. That's, I think,
Yeah,
translatability from a big picture is really that.
absolutely. And I think that awareness of the not just the need to reach more diverse populations, but the fact that different populations have different risk factors, which they may have been unaware
Right.
of, is such an important step that we've been taking in the past few years, but it is quite
Yes.
recent. And in fact, it was the peers and the lead of one of the peers back in 2018 who really pioneered the working paper, which has been very impactful. I guess you're familiar with it, but it was really sort of saying, look, we can't just carry on
Right.
because we know there are these differences between groups, and we should sort of combine our strengths to raise this awareness. And I think that's been a real turning point in the past 5 years, and it's great to hear that you're sort of on the sort of front line of that in some ways.
Yeah, absolutely. And I think that echoes the same sentiment. If we discover something in a mouse, but it's not happening in a human, it's not that important. And the same concept happens if we discover things happening in only predominantly non-Hispanic white populations. How can we say this is happening truly in other
Yeah,
populations? So I agree, absolutely.
yeah. And I guess the other side of that is the risk reduction efforts,
Mhm.
which again are also really elevated in the past sort of 10 years or so, and targeting those more precisely to different populations who have those different risk factors.
Yes, absolutely.
It's also such an important part of the effort, right?
Yeah, absolutely. I think I recently heard about a really interesting effort in risk reduction, specifically out of my alma mater, UC Davis, the ADRC there. They have a really large Asian American population. So they helped create a community-based exercise program that harnessed different Southeast Asian physical activity practices. So they had this whole rotational program with instructors to bring to the community that was culturally relevant and important. So I think that is, a key feature to
Yeah,
building those trusts. Yeah.
absolutely. And so your involvement with ISTAR is through the Immunity and Neurodegeneration peer.
It is.
Yeah,
Yeah.
yeah. Well, how long have you been a member of that?
So I started as a student representative in 2022, and then I just got re-elected to do the next 2 years until 2026, um, starting after AAIC this year.
Mhm. Right. And so for the listeners, can you tell us a little bit about the focus of that peer?
Yeah, absolutely. So ARPIA is really aimed to talk about the different immune cells in the body, both centrally and peripherally. And I focus in the central nervous system, but I think we do an excellent job of bringing both peripheral and central nervous system. And we know, based on recent research, that the cross section and the interactivity of those cells in the spinal cord, at the blood-brain barrier, and at the surface of the brain are super important in risk and resilience to Alzheimer's and infection in general. So our goal is to harness the expertise of our PM members and our executive committee, who are experts in the field of immune cells and neurodegeneration, and look at it from a variety of perspectives. We have exercise biologists, we have circadian rhythm specialists, we have kind of degenerative disease pathologists that kind of focus on the tail end of disease. We have immune activation. And really, our goal, I think, is to build a really inclusive environment to foster conversations, easy flow of ideas and activities. And then we have a very active roadmap working group, which is I'm not sure if all the PIAs have it. I'm not as familiar with the whole hierarchical structure, but
Yeah.
we have a very active roadmap working group, which is basically a group of people that are in our PIA that are interested in kind of consolidating information and writing review articles or kind of articles that help maybe on a particular technique, what we should be thinking about when we're looking at microscopy images or something like that. And then publishing those so that everybody can have access to those recommendations or those review articles. And I wrote a note. So our working group has published 5 papers on some really interesting topics, including reactive astrocytes, knowledge gaps in Alzheimer's disease and immune biomarkers, central and peripheral immune system crosstalk, the impact of COVID-19 on the onset and progression of AD. That one is a really good paper. And the diversity of transcriptomic microglial phenotypes in aging and Alzheimer's disease. So kind of from the name of those, you can hear that there's a vast variety in the things that we do. But I think that's the really awesome part of our PIA is that we do have such a diversity in expertise that the conversations I feel are very productive because we can provide kind of insight on one problem from a variety of different aspects, which I've really enjoyed.
It sounds as if you're able to combine multiple different perspectives at different levels of investigation, really, and that seems to be symbolizing a lot of the way research in this field is moving forward. It's breaking down the silos and bringing together those different researchers, and it's really fascinating to hear how you're doing that with the immunity cells. So what are some of the hot topics? You just told us some of the publications. So what are some of the real hot, exciting topics at the moment? What, that we might expect to see at AAIC, for example?
I'm actually going to refer back to our year in review webinar. So the PIAs all have the option to host a year in review webinar in January, which essentially one of our members gets to talk about really cool research that's happened in the last year and things that we're looking forward to. So our communications chair, Dr. Wade Self, did an excellent job in January talking about some really hot topics. So I'm going to refer back to those. But I think, and I can definitely speak on this one, I think the epidemiological and genetic links between immune reactivity and Alzheimer's disease pathogenesis is a very hot topic, which I've also been interested in my research. Genetic variants, absolutely, and factors outside just the brain that modify multifaceted microglial activation. So peripheral genetic factors that influence central nervous reactivity, which is very intriguing. Innate and adaptive immune mechanisms that drive neurodegeneration, and how immunity influences the outcome of anti-amyloid therapeutics. So there have been a lot of publications recently on how our innate and peripheral immune system react to anti-amyloid therapies. And then generally, how can we improve neuroimmune biomarkers? That's a really difficult one because immune cells are circulatory cells in the periphery. So being able to adequately, surveil the immune cells in the blood and relate that directly to Alzheimer's disease, that's a really difficult topic. And we know accessing circulating cells in the brain often is like a spinal tap, which is fairly invasive and not really a great way to just see what the inflammatory profile might be. So improving mechanisms of doing that.
Right. So there's quite a lot going on in this topic because, in a way, it's one of the aspects that's been perhaps going on the longest. If we think back to the original sort of Alzheimer's original case presentation, and I recall you said earlier on about your work is also interested in postmortem brain tissue examination, which is slightly different to what you were just describing. But I'm wondering then, given that people have been doing that for more than 100 years, what sort of questions are you asking when you're doing that aspect of your own research?
My area of expertise. So I think specifically looking at neuroimmune in postmortem human AD tissue. Specifically, I think a hot area that I'm also working on is specifically characterizing cell populations with a regionality specificity. So that basically means what types of microglia, what cell surface proteins and receptors are they expressing close and distal to different pathologies. And that, in my opinion, is a key distinguishment between just basic kind of postmortem diagnostic criteria of, yes, it's Alzheimer's, there's amyloid, and there's tau, and it's throughout the brain. Awesome. But that is just one tiny feature. Yeah, it's a pathological diagnostic criteria, but the neuroimmune cells, the astrocytes, the microglia, the oligodendrocytes, what are they doing? What's distribution and what surface markers are they expressing? And in my research, I'm really interested in specifically post-translational modification of microglia. So microglia have dynamic activation states. So they should shift between homeostatic, resting, just surveilling the brain, eating up dead things, debris, things like that. But in Alzheimer's disease, they have this kind of shift into activated state or reactive, and then they're unable to resolve that. And therefore, they will be more neuroinflammatory, have more expression and release of cytokines and chemokines, and create that kind of local neuroinflammatory environment, which we know that's awesome. But what are the receptors and modifications on their cells that specifically cause that lack of resolution of reactivity states? And my research has focused on that, and it's been really cool. I found it first in mice, this really interesting post-translational modification. And we said, okay, it's awesome in mice. Is it happening in humans? Oh, it's happening in humans. It's happening in a very interesting regionality, proximity to amyloid beta plaques. Okay, what does that mean? So we've kind of dug deeper into understanding it. And now we're working on amyloid-laden vessels. So I'm looking at cerebral amyloid angiopathy and how microglia react to that. So it's been really fun.
So are there any particular sort of recent, I don't know about breakthroughs, but sort of availability of tools and technologies that have made this type of investigation possible? And what sort of things are you using?
Absolutely. So in my opinion, one of the coolest tools that I've been using is single cell RNA sequencing. So finding a cell population, and then a lot of major, huge studies will have hundreds of Alzheimer's disease brains, and they'll take regions of the brain like the frontal cortex, or the hippocampus, or the temporal cortex, and they'll do single cell sequencing, and they'll come up with all of these markers that are theoretically expressed on different kind of states along the spectrum of different glial cells, astrocytes and microglia, and oligodendrocytes. I focus mostly on microglia, but basically what we've been trying to do is find these very unique but intriguing markers that are on disease-associated cells, and then localize those to the protein level in our tissue, and then use what's called RNA scope, where I can probe the RNA level and then find it regionality with the protein marker. And that's currently what my thesis work, kind of the tail end of my thesis work is doing, is to identify very intriguing markers and then provide regionality, specificity in the tissue architecture. So that's a really unique feature of using tissue, is I can say it's in this cell, it's in this layer of the cortical tissue, and it is this close to X pathology. So in my opinion, there are limitations with postmortem human tissue. We can't manipulate it very much, but we can do some really intriguing investigations with hypotheses that are, I think, equally as valid, just in a different approach. And then a lot of times when I find these cool markers, I can go back nicely to my cell model and I can say, okay, let's see if we treat it with a drug that blocks this, what happens? Or I can do, I can isolate microglia from an Alzheimer's disease mouse model, and then I can sort those based on cell surface expression of different receptors and say, oh, this population is expressing X, while this population is expressing Y. And that's the nice thing. I can find cool things in humans and then go back to my model systems and see if they're recapitulated in a model system, which has been a godsend in my thesis work. Yeah.
So in terms of then the potential for applications in the future, or connecting this to people who might be at risk of developing, say, Alzheimer's or some other neurodegenerative condition, where do you see what you're doing right now? Where do you see, or where do you hope it might be going?
It's a little tricky with the postmortem stuff, only because I can only speculate based on what I'm seeing and, post life. But based on what we're seeing, it appears that there are definitely some non-pharmaceutical or pharmacology interventions early in life that appear to reduce risk of cardiovascular disease and atherosclerosis in the brain. And we know when vessels get really, really hard, there's actually likelihood of a leakage of amyloid that is being cleared from the brain through the CSF or interstitial fluids. and blood that then become kind of into the brain. So if you improve your cardiovascular health, reduce your risks of athero and arteriosclerosis, you're probably putting your brain in a better position to not leak pathology essentially into the cortex. But in terms of my kind of biomarker approach and my investigatory approach, there actually are a couple drugs that specifically target my PTM of interest that are in early clinical trials in I think Alzheimer's disease, but also in a tauopathy model. Tauopathy, I think it's progressive supranuclear palsy. But it's early phase clinical trials. But I think that's cool because you can say, oh, I've identified this interesting thing. Other researchers are also identifying a very interesting modification that appears to potentially control some aspect of microglial activity. And now they're seeing if inhibition of that would be beneficial in terms of neuroinflammation in the brain. I guess another component of that is other researchers at our institution have identified really intriguing kind of protein folding like scaffolds. It's not my expertise, but they've identified different ways that proteins fold and how those kind of DNA components are translated and transcribed. So they also have a clinical trial going to see if inhibition of a certain modification or folding structure can improve health span in humans. So I think it's cool because you can look at a basic science perspective and still have something that's relative and meaningful to humans. It just takes a really long time to get there.
Yeah, but it's really important though, isn't it, for that sort of encouraging people not just to participate in studies, but to donate their brain, for instance, or to engage with some of these lifestyle modifications that could change their risk. So having that information, that data, and being able to visualize it in some way and show them, I think is really important. And it sounds like that's sort of where you're positioned. Yeah. And are you presenting any of this at the AAIC?
Yes. So I'll be presenting my research at a poster session. I think it's on July 28th. I'll be presenting some of my mouse and cell culture systems, as well as a little human stuff, which will be fun. But I also this year am a ISTAART ambassador. So I'm part of the cohort of students and postdocs that essentially we wear the purple shirts and we'll be running around helping the sessions with the session speakers, helping moderate sessions. And I actually will also be speaking on an ISTAART panel. Oh gosh, I think it's on Wednesday, the 31st, about leveraging student engagement and activism in ISTAART. So how we can utilize students and our interest in being involved in scientific societies and kind of tie us in with local Alzheimer's chapters to see if there is some synergy between institutions and Alzheimer's chapters.
You're obviously a very, positive advocate for research, but also it sounds as if your involvement with ISTAART and the PIA have also are really important for your progression as a researcher. So would you have any messages for other sort of people who are just starting out in, say, their doctoral research? What would you say to them?
Yes, absolutely. I would say, and this is very transparent, when I started my graduate program in 2020 in the pandemic, I was living alone across the country, away from everybody. And I said, oh, I am not happy. I need something that I can really put my passion to work and feel like I'm driving myself outside of just research alone. And my faculty advisor is involved in the Immunity PIA, and she said, Caitlyn, you really like to talk and you love collaboration. So I think you should get involved. And at first I was very nervous because I thought, I'm a trainee, I'm a second year PhD student. Nobody's going to take anything I say seriously. And actually, it turns out it doesn't really matter what level of training that you're at. It's about your attitude. And if you're open to learning and talking and listening, you will learn so much about not only the research, but how to interact with other scientists and build good quality connections that will help you. in terms of if you need resources for a research project, expertise on grant writing, or how to apply for particular grants. Most likely somebody in your PIA will have done that in their past life. But also the important part of being involved in the ISTAART and Alzheimer's Association communities is that it takes you as one little dot on the map and puts you with hundreds of thousands of other dots on the map. So to know that you're not alone and likely not doing research that's totally different than somebody else. There's people doing similar things. There are people that you could bounce ideas off of, learn things, collaborate. Last year at AAIC in Amsterdam, I met a bunch of students that were really interested in writing. I'm really interested in writing. It was an awesome opportunity to talk about how do you write a manuscript? How do you write a grant? How can I ask my advisor to be more involved in the writing process if that was a barrier of access? So I think it also made me more confident. Like, I can hold my own. I can have a conversation, obviously with understanding that I'm not an expert, but I'm an expert in my own stuff, and I can kind of hold my own in those conversations and provide valuable insight and input. I'm not just a trainee. I'm a trainee that also has kind of cool things to say. And I like that I feel appreciated in the ISTAART community. Absolutely.
It's so good that it offers those opportunities, as you said, for people at every stage. You don't have to have achieved certain things before you can be included, that it's actually it is very inclusive for everybody. And I think there are a lot of activities throughout the year, as well as at the conference itself, which are focused on early career researchers. And I think that I agree that is one of the strengths, and that being welcoming to that enormous community is such a great aspect of ISTAART, and just really getting people involved, which is wonderful. So this has been really so fascinating, both hearing about your research and the work of the PIA. But it is time to end today's podcast. But before we go, I do have a final question. So why should listeners sign up for your PIA, the Immunity and Neurodegeneration PIA?
Yes, thank you for the great question. I think if you're a scientist at any training level and interested in the cross-section between both central and peripheral immune system and neurodegenerative diseases, you would really enjoy being part of our PIA. I think we're a very dynamic group of scientists interested in microglia, T cells, and other exciting intersections of immune cells. And we also welcome all perspectives and ideas. So I think if you like those topics, you would really enjoy being part of our PIA.
Great. Well, I'd like to extend my heartiest thanks to Caitlyn for taking the time to join us today. And thank you to everybody who is listening. You can find profiles of myself and my absolutely wonderful, passionate, excited guest, and information on how to become involved in ISTAART on our website at dementiaresearcher.nihr.ac.uk, and also at owls.org/ISTAART. And there will be a link in the show notes. I'm Arlene Astell, and you've been listening to the Relay podcast from Dementia Researcher and the Alzheimer's Association. Hit subscribe on YouTube or in your favorite podcast app to ensure you don't miss an episode. Thank you and goodbye.
The Dementia Researcher podcast was brought to you by University College London with generous funding from the UK National Institute for Health Research, Alzheimer's Research UK, Alzheimer's Society, Alzheimer's Association, and Race Against Dementia. Please subscribe, leave us a review, and register on our website for full access to all our great resources. dementiaresearcher.nihr.ac.uk.